The impact of O2 availability on human cancer. This was printed in the National Library of Medicine, National Institute of Health, in the Howard Hughes Medical Institute. Abstract. During the last century, it's been established that regions within solid tumors experience mild to severe oxygen deprivation due to aberrant vascular function. These hypoxic regions are associated with altered cellular metabolism as well as increased resistance to radiation and chemotherapy. As discussed in this timeline over the past decade, work from many laboratories has elucidated the mechanisms by which hypoxia-inducible factors modulate tumor cell metabolism, angiogenesis, growth, and metastasis. The central role played by intratumoral hypoxia and HTF in these processes has made them attractive therapeutic targets in the treatment of multiple human malignancies. Oxygen O2 is required for aerobic metabolism to maintain intracellular bioenergetics and serve as an electron acceptor in many organic and inorganic reactions. Hypoxia, defined as reduced OT levels, occurs in a variety of pathological conditions, including stroke, tissue ischemia, inflammation, and growth of solid tumors. The beginnings of hypoxia research in tumor biology can be traced back to observations made in the early 20th century by Otto Warburg, who demonstrated that unlike normal cells, tumor cells favor glycolysis independent of cellular oxygen levels. He postulated that tumor growth is caused by mitochondrial dysfunction in neoplastic cells, forcing them to generate energy through glycosis. This hypothesis appears to be incorrect, but a number of other molecular mechanisms promoting aerobic glycosis has been proposed through mutations and epigenetic changes in genes encoding tumor suppressors, oncogene activation, and hypoxic adaptations. Ambient air is 21% O2. However, most mammalian tissue exists at 2% to 9% O2 on average. Hypoxia is usually defined as less than 2% O2, while severe hypoxia or anoxia is defined as less than 0.02% O2. In the decades following Warburg's observations, scientists sought to determine whether hypoxic or anoxic cells could be found in mammalian tumors and how these cells affected radiation therapy. Of great interest to radiation biologists and oncologists, the radioprotective effects of anxiety in normal tissue was demonstrated in 1940s by Lacoga and Evans and others, using whole-body anoxia in newborn rodents. If a subset of tumor cells did in fact exist in an environment deprived of O2, then they might be responsible for tumor recurrence after radiation. This realization is still fueling research today, over 60 years later. Early demonstrations of tumor hypoxia. Before hypoxic cells could be visualized as tumors, their presence was inferred by some astute observations. In 1955, Tomlinson and Gray studied histology sections of human lung tumors and observed cells growing in cords, running parallel to vascularized stroma. In large cords, they noted a necrotic core surrounded by a region of visual cells neighboring a capillary vessel. They proposed that necrosis was due to insufficient O2 and nutrients supply to the rapidly expanding tissue. Regardless of cord size, detectable bands of live cancer cells lying between the necrotic core and the surrounding stroma were consistently 170 microns in width, approximately the calculated distance of O2 diffusion. They propose that the edges of such necrotic cores harbor viable hypoxic tumor cells. Other human tumors, such as cervical and renal carcinomas, exhibited similar histological characteristics, suggesting that this was not unique to the lung. The histological specimens also suggested that tumor cells were exposed to an O2 gradient ranging from efficient oxygenation near the stroma, decreasing gradually to near anoxia bordering the necrotic regions. These varying O2 concentrations have since been shown to have significantly different effects on cellular processes. Further evidence for the existence of viable hypoxic tumor cells that may influence tumor responses to radiation was offered by Powers and Tomic. They irradiated lymphosarcomas in mice breathing either ambient air or hyperbaric O2, 3 atmospheres of pressure, before transplanting them to a new mouse host. They observed decreased tumor cell survival if transplanted from mice breathing hyperbaric O2 as compared to 21%. Suggested that certain tumor cells were protected by decreased O2 conditions, but sensitized by a surplus of O2 delivered to the host. Moreover, tumors irradiated in dead mice, likely more hypoxic than viable animals breathing room air, were much less sensitive to radiation. Around the same time, Churchill-Davidson made similar encouraging observations in cancer patients treated with radiation in combination with hyperbaric O2. While these results were promising and suggestive of the existence of hypoxic cells within tumors, they did not provide direct evidence, and it was not until the later decades of the 20th century that precise techniques for measuring O2 levels were developed. Even today, scientists lacked an optimal method of direct tissue O2 measurement that is non-invasive, precise, and quantitative. But this goal is clearly within reach. In the 1980s, in vivo polarographic measurements with electrodes and in vivo cyrospectrophotometric measurements of oxyhemoglobin provided some insight into the regional oxygenation status of tumors. Nuclear magnetic resonance spectroscopy of P signals detecting tumor cell metabolism and sensitizer optics such as mesondazole selectively binding hypoxic cells were also used in an attempt to identify hypoxic regions. However, none of these methods were ideal. While electrode measurements were certainly the most direct form of O2 measurement available, some human tumors were not accessible to electrodes, and the electrode diameters were too large to allow precise measurement of cellular oxygenation status. Instead, they likely measured average O2 tensions in tumor sections, which would not accurately reflect dramatic variations that occur over very small distances. Cyrospectrophotometric measurements readily determine the oxyhemoglobin saturation of individual erythrocytes in tumor cells ex vivo, an indirect assessment of tumor oxygenation. PNMR spectroscopy was found to lack sensitivity, and technical difficulties still limited the use of sensitizer adducts. Using several of these methods, studies in both the 1980s and the early 1990s found decreased average O2 tensions in human tumor tissues as compared to normal tissue. As predicted by previous work, Substantial intertumoral variability was observed, however, indicating that predictions of tumor oxygenation would be difficult and underscoring the urgent need for more precise and direct O2 measurement techniques easily used in clinical settings. In the late 1980s, an additional complication was revealed by Chaplin and others using the Horsch 33432. This DNA binding stain has a short distribution half-life but remains bound to tumor cell DNA, thus labeling cells based on their proximity to the blood supply. Using this technique, coupled with cell sorting and responses to treatment, Chaplin differentiated between diffusion-limited chronic hypoxia and acute hypoxia, resulting from transient changes in blood flow through tumor vasculature of larger tumors. Cells adjacent to blood vessels may suddenly and intermittently become hypoxic as the vessel becomes momentarily occluded during treatment. These two types of hypoxia may have different effects on treatment responses, a possibility that must be considered in the selection of cancer therapy protocols. The effect of hypoxia on cancer. As described by Otto Warburg in the 1920s, rapidly dividing tumor cells display increased glycolysis even in the presence of oxygen. As a consequence, lactic acid concentrations are elevated, acidifying the environment. Initially, scientists realized that exposure to hypoxia slowed tumor growth in mice, although early experiments were confounded by hypoxia-induced generalized weight loss in experimental animals. In 1954, Barrick and Brickerman repeated these experiments in hypothyroid and acclimatized mice and found similar results without generalized weight loss, confirming that O2 deprivation impaired tumor growth. In extreme cases of prolonged anoxia, cells would undergo necrosis, as noted in Tomlinson and Gray's tumor sections. The major focus of hypoxia research for most of the 20th century was its role in cancer treatment responses. As early as 1909, Schwartz and colleagues noted that normal mammalian cells irradiated under conditions of hypoxia or anoxia were less sensitive to radiation than those irradiated in the presence of O2. At that time, the changes were attributed to blood flow differences and not to oxygen availability. In 1923, Petrie's studies on the effects of oxygen on vegetable seeds were the first to note a correlation between oxygen and radiosensitivity. In the 1950s, pioneering work by Gray and his colleagues established that existence of an hypoxic radioresistance effect in mammalian tumors and sought to quantify it. Gray used Ehrlich mouse ascites tumor cells irradiated in vitro and in mice under various O2 conditions. Similar to what has been shown in plants and insect tissues, Gray found that the ascites tumor cells showed a dependence of radiation sensitivity in O2 tension both in vitro and in vivo. Sensitivity was about 3 times greater under well-oxygenated conditions as compared to anoxic conditions. Subsequently, Churchill-Davidson published a cytological evaluation of damage in patient tumors after radiation. One half of the tumor was irradiated while patients breathed pure O2 at 3 atmospheres pressure. The other half while patients were breathing room air. Significantly more damage was observed in the half irradiated under high O2 oxygen conditions. In 1960, Dewey used human embryo liver cells to show that cells grown in vitro had under anoxic conditions exhibited approximately 2.5-fold greater colony forming ability after irradiation than cells grown in air. Harrod and Wilson irradiated mouse leukemia cells in live mice or 2 minutes after death and observed an identical radiosensitivity ratio as observed by Dewey. Several years later, the experiments by Powers and Tomic described above further confirmed these findings. Numerous other studies followed evaluating hypoxic effects on transplanted or spontaneous tumor regression after irradiation in mice, confirming earlier experiments. Similarly, hypoxic cells were later found to be more resistant to commonly used chemotherapeutic agents. However, the 1970s and 1980s witnessed a shift in opinions concerning the importance of hypoxia in clinical responses to cancer treatments. Firstly, clinical responses to hyperbaric O2 treatment and later to hypoxic cell radiosensitizers were disappointing. Furthermore, hypoxic tumor cells appeared to reoxygenate in response to fractionated radiation treatment, and further would be substantially less impact on tumor cell responses after fractionated therapy than after a single radiation dose. Finally, scientists found that intrinsic cellular radiosensitivity alone at low doses could explain the differences in clinical responses observed. Many became skeptical that the radiosensitization of hypoxic tumor cells was going to be clinically beneficial. The next decade witnessed a second shift in conventional wisdom, with a demonstration of the 1980s and 90s that, like other mammalian tumors, human tumors also contained radio-resistant hypoxic cells, came the realization that hypoxic cells could be targeted directly with hypoxia-specific cytotoxins. The combination of radiation and hypoxia-specific cytotoxin would thus have the potential to destroy the entire tumor cell population. Such cytotoxins thus became the focus of intense investigation in the early 1990s. The question of the role of hypoxia in cancer treatment instigated many studies, some of which revealed a number of other critical effects of hypoxia on tumor progression, as discussed in detail later, including angiogenesis, metastatic potential, DNA replication, and reduced protein synthesis. Finally, in the decade immediately preceding the discovery of the transcription factor, hypoxia-inducible factor, the expression of a number of genes was determined to be stimulated by O2 deprivation. The earliest to be identified were glucose-regulated proteins and O2-regulated proteins. One ORP was later identified as heme oxygenase, while two others were revealed to be the same as 2 GRPs. Other genes induced by hypoxia include those regulating hematopoiesis and vasculature, and erythropoietin glycolysis metastasis, and DNA damage responses. How the cell sensed O2 deprivation and how gene transcription was regulated by hypoxia were completely open questions until the discovery of HIFs in the 1990s and further elucidation of HIF-independent effects of hypoxia on cellular processes over the next decades. Identification of hypoxia-inducible factors, HIFs. HIFs are heterodimeric transcription factors consisting of two subunits, systematic characteristic of the EPO gene by multiple groups led to the identification of a cis-acting hypoxia response element. in the 3-foot flanking region of this locus that confers O2 regulation of EPO expression. HIF1 was subsequently described by Greg Semsa and Guang Wang in 1992 as a nuclear factor that was induced by hypoxia, binds to the HPO HRE, and promotes transcriptional activation of HPO in O2-starved cells. The HIF1 binding site within the HPO HRE was used for its purification via DNA affinity, chromatography, and cloning of DNAs encoding the HIF1 and HIF1 subunits in 1995. This demonstrated that both subunits are members of the basic helix loop helix polypeptide family that contain a PAS domain, initially characterized in the Drosophila melanogaster peer and sim proteins and mammalian aerohydrocarbon nuclear translocator. Because MRC is commonly dysregulated in cancer, this is also an important interaction to consider when developing anti-cancer therapies. Therapeutic implications of tumor hypoxia. As described previously, early work on the role of O2 levels in cancer centered on the observation that poorly perfused tissues are more resistant to radiation. It was later that Gray made the direct connection between tissue oxygenation and tumor cell radioresistance, a principle known as O2 enhancement effect. Oxygen was thought to act as a direct radiosensitizer, increasing damage to DNA through the formation of free radicals. Indeed, experiments conducted by Richard Hotchkiss and colleagues in the late 1980s demonstrated that the oxygen effect required O2 to be present at the exact time of radiation, leading to the oxygen fixation hypothesis. The relative contribution of cellular responses to hypoxia reverse the direct effect of free radicals to hypoxic radioresistance is still being deciphered. Box 2, the oxygen fixation hypothesis. The irradiation of cells generates free radicals either in DNA or water molecules. Free radicals in DNA can react with available oxygen to generate a peroxy radical DNA-OO, thus chemically modifying the DNA, oxygen fixation. In the absence of O2, the DNA radical will be reduced, restoring the DNA to its original composition. In addition to the direct effects on DNA, free radicals produced from H2O can further damage nearby DNA. Early attempts to exploit the oxygen effect for therapeutic benefit focused on interventions to increase tumor oxygenation during radiation. Hypobaric O2 red blood cell transfusions and erythropoietin administration were all attempted as attempts to increase tumor oxygenation. However, these approaches did not gain widespread use due to conflicting reports of their efficacy in clinical trials. In the 1970s, nitroimodazine derivatives, which act as molecular mimics for O2, were tested in conjunction with radiation. Despite initial promise, clinical trials with nitroimodazines demonstrated limited benefit, in part due to dose-limiting toxicities. As an alternative to increasing tumor oxygenation, more recent strategies have attempted to take advantage of hypoxia to selectively kill tumor cells. Tirapirazine, described over 20 years ago, is a prototypic example of hypoxia-activated prodrug. Terrapromazine is specifically reduced to hypoxic cells, forming radical species that poison topoisomerase II, leading to DNA double-strand breaks. Clinical trials with this agent have demonstrated benefit in patients with lung as well as head and neck cancer when used in combination with radiation or chemotherapy. In addition to the direct role of molecular O2 in generating radiation-induced DNA damage, the biological effect of hypoxia on tumor cells can also modulate their response to therapy. This was first demonstrated several decades ago in experiments showing that the duration of hypoxia influenced radiation sensitivity. Collectively, hypoxia modulates multiple pathways that contribute to radiation and chemotherapy resistance. Hypoxia exerts a selective pressure on cells for loss of p53, a key mediator of apoptosis, and upregulates the expression of the multi-drug resistance gene, leading to efflux of chemotherapeutic drugs. Hypoxia has also been shown to contribute to increased mutagenesis rates and metastases. These are examples of genes directly regulated by hypoxia that play important roles in metastases and could serve as druggable targets. Given the impact of hypoxia on therapeutic efficacy and patient prognosis, the ability to measure and image hypoxia in tumors has garnered significant attention. The introduction of the polarized O2 microelectrode in the 1990s allowed the direct and most accurate measurement of O2 tension in tumors. However, due to the invasive nature of this technique and limitation to superficial tumors, it cannot be broadly utilized in the clinic. Thus, the emphasis has shifted to imaging modalities to indirectly observe tumor hypoxia. Increased glycosis by tumors can be imaged using positron emission tomography with fluorodeoxyglucose as a tracer. However, since many tumors display increased rates of aerobic glycosis, this is not a selective marker for tumor hypoxia. Several other compounds have also been developed that selectively accumulate in hypoxic tissues, including fluoromazidazole, eterodazole, pentafluoride, and cu-diacetyl methylsimicabazone. Nitroimidazoles are reduced under hypoxic conditions, forming stable adducts in the cell that can be detected by the PET scanner. While many of these approaches are currently being investigated for their ability to predict treatment response, they still have not attained routine use in the clinic. The cloning of HIF, understanding of the pathways that regulate it, and identification of downstream HIF targets has produced numerous molecular targets for therapeutic intervention. Targeting the HIF pathway has generated significant attention because of the potential to selectively target hypoxic tumor cells and the role that hypoxia plays in key tumorigenic processes such as metastases, angiogenesis, and metabolism. Due to the difficulty in directly targeting transcription factors, the most promising intervention includes strategies that block HIF accumulation, either by inhibiting translation or promoting degradation. Another approach is to inhibit key HIF target genes such as VEGF, TGFA, and the LYSLY oxidase. Mutations in the P13K, AKT, mTOR, or RAS signaling pathways are commonly found in human tumors, and both of these pathways have been shown to upregulate HIF protein. Indeed, inhibition of the mTOR kinase and RAD001 in the mouse model of prostate cancer induced by oncogenic AKT was found to decrease HIF accumulation and HIF-dependent gene expression. Renal carcinoma cells, which often exhibit mutations in the VHL tumor suppression, have also been shown to downregulate HIF expression in response to TOR inhibition. In addition to the hypoxia-regulated VHL-dependent HIF degradation pathway, HIF is degraded through interaction with receptor of activated protein kinase. RACK1 competes with the HSP90 for binding to HIF1 and HSP90 inhibitors and have been shown to promote HIF degradation. Targeting the interaction between the HIF and the transcriptional co-activator P300 also been demonstrated to inhibit HIF target gene expression and impair tumor growth. Numerous other small molecules, including topoisomerase inhibitors, microtubular destabilizers, and histone deacetylase inhibitors, inhibit HIF accumulation, although the mechanism for these effects is not yet clear. Finally, drugs against several key HIF transcriptional targets have been developed and approved for clinical use. Chief among these is bevericizumab, Avastin, and Genentech, a monoclonal antibody against VEGF, a key endothelial growth factor involved in tumor angiogenesis. Bevericizumab is now commonly used for the treatment of metastatic colorectal cancer. VEGF activity is also shown, can also be inhibited by VEGF receptor tyrosine kinase inhibitors, such as sorafenib, which has been shown clinically to benefit in renal and hepatocellular carcinoma. The therapeutic effects of anti-VEGF therapy appear to involve several mechanisms, often depending on the tumor type and chemotherapy regimen. These effects include inhibition of angiogenesis, epithelial cell apoptosis, and vessel normalization. Additionally, epidermal growth factor receptor inhibitors such as ERISA have seen some success in the treatment of lung cancer. The past century of hypoxia research has provided significant advancement in our understanding of the molecular pathways by which O2 tension influences the properties of tumors, from proliferation and angiogenesis to radiation and chemotherapy resistance. Current studies are focused on extending observations made in vitro to more physiological contexts. The cloning of the HIFs has provided a genetic tool that, when coupled with established mouse models of cancer, will help clarify HIF-dependent effects on disease progression. Accordingly, studies in which one or both of the HIF subunits is conditionally deleted are now underway and will allow us to determine the tissue and stage-specific effects of these factors on tumor progression. Mouse models will also be a useful platform to test newly identified therapies that target the HIF pathway for the treatment of cancer. The recent identification of a number of hypoxia-regulated microRNAs provides another mechanism by which O2 levels can influence gene expression. A number of these microRNAs are highly expressed in tumors, and their predicted targets have roles in apoptosis, angiogenesis, and proliferation. In future studies, it will be important to identify the targets of these microRNAs to understand their role in tumorigenesis. Nearly a century after Warburg's initial observation of aerobic glycolysis in tumors, the unique metabolism of cancer cells will be important to re-examine, specifically the crosstalk between the HIF pathway and dysregulated oncogene and tumor suppressor function in driving metabolic changes will be an important area of study. Collectively, these observations will be critical for the successful development of novel imaging and treatment modalities, with the promise to improve targeted therapy of cancer and overcome therapeutic resistance induced by hypoxia. Box 1 timeline. The history of research on tumor hypoxia. 1901. Schwartz first observed the effects of changes in vascular function on radiation sensitivity. 1923. First correlation between oxygen and radiosensitivity made by Petrie in Germany. 1924. Otto Warburg describes the increased tumor cell glycolysis in the presence of high OT2 levels. 1953. Gray and colleagues established the existence of hypoxic radioresistance. 1953. Tomlinson and Gray described tumor cell hypoxia in clinical specimens. 1964. Hypobaric O2 used to promote the efficacy of radiation treatment in cancer patients. 1984. First oxygen-regulated proteins described. 1986. Polarizographic electrodes and nuclear magnetic resonance spectroscopy used to measure tumor cell oxygenation. 1986. First attempts to exploit tumor cell hypoxia by selectively killing O2-starved cells with teroprazimazine. 1991. Hypoxia-regulated DNA elements characterized in human erythropoietin EPO gene. 1995. Hypoxia-inducible factor 1, HIF1, biochemically purified and cloned by Semenza and colleagues. 1996. Garcia and co-workers determined that tumor cell hypoxia selects for rare clones exhibiting p53 inactivating mutations. 1997. First demonstration of tumor-promoting effects by HIF in subcutaneous mouse models. 1999. HIF regulation by von Hippel-Lindau tumor suppressor protein, PVHL, described by Ratcliffe and others. 2001. Ratcliffe, Kaline, and Lee research groups demonstrate HIF subunit proline hydroxylation. 2001. HIF proxyl hydrolase domain PHD enzymes identified. Box 3. HIF contributions to other diseases. Polycythemia. Point mutations in HIF-2 alpha have recently been identified in multiple families with erythrocytosis A point mutation in VHL leading to Truvash polycythemia preferentially stabilizes HIF-2 alpha. Cardiovascular disease. Cardiac ischemia is a significant source of patient mortality. The adaptive responses mediated by the HIFs are important for protecting ischemic tissues from injury and activating the HIF pathway selectively in these tissues could provide an important therapeutic intervention. Pulmonary hypertension. Both HIF-1 alpha and HIF-2 alpha regulate vascular remodeling and development of pulmonary hypertension in response to chronic hypoxia. Inflammation. Hypoxia is commonly observed in inflammatory states, and HIF-1 alpha can have protective as well as pro-inflammatory effects. Macrophage-specific deletion of HIF-1 alpha revealed an important role in mediating innate immune responses. Alternatively, deletion of HIF-1 alpha in colonic epithelium revealed a protective function in drug-induced colitis.